Susie,
Rob,
do you know what an animal is and what an animal is not?
if we think back to sort of like, that thing when you like, was it GCSE biology or something? There was that,
Hmm.
was it Mr. Green or something? There was an
Mrs. Gren.
acronym. Mrs. Gren.
That was
It was
that was
like,
all life. I guess
that was
that's
life.
things respiring and eating. But what is an
Yes,
actual
okay.
animal? That's what
Oh,
I want to get at for this today's episode. But before we go down that, I guess I'm curious as to what your initial. I mean, as a dinosaur person, you don't spend a long time thinking about that. But what is an animal?
actually, it's a good point because, I'm going, oh, well, it's got to have a head, but then there's loads of animals that don't have
Yep.
heads. And then it doesn't have to have a brain. Do
Nope.
sponges have brains? Do corals
Nope.
have brains?
Nope.
Oh my God, Rob, what's an animal? I think I might, as this, I'm going to have a crisis.
Well, the bigger question would be, we don't. I mean, this is difficult because we can't actually agree what an animal is and what an animal is not. Therefore, what would it even look like 550 million years ago when they first turned up? That's the real challenge.
Well,
And
and how
that's
would we tell?
and
Because
how
they're
would
going
we
to be
tell?
squashed
They're going to
over
be
under
squishy,
600 million years of rock.
and that's exactly what we're going to talk about today. Okay, so Susie, since we last met, we asked our lovely Fossil Files listeners to vote on what they wanted us to do for our next choice of topic, because every week you and I will talk about a particular paper, and we've done all sorts of different things. What are your highlights that you can think of off the top of your head?
Well, can I just ask, first of all, did both
Yeah.
our listeners vote or only one of them?
Did you vote? I don't know.
I
I
didn't vote.
I didn't vote either, but I felt like that would be cheating because I had my own preferences about what I wanted to happen. So in the past, we've done plesiosaurs, early vertebrates, fossil fails, dinosaurs on the moon, all sorts of
Oh,
things.
that was my favorite. That was my favorite. I
Fossil
really enjoyed
trackways,
I
pterosaurs,
know
Neanderthals.
the dinosaurs on the moon. I know that was only a side note, but I think that was my highlight as a paper.
Yeah, that if you haven't listened to the dinosaurs on the moon, go and check that out. But I put together this list of things for people to choose from, and I was firmly expecting people to choose the Triassic dinosaurs because I thought everyone would love dinosaurs. Wrong. Paleozoic ones, early animals came position number one and two in the poll. So
Wow.
our winner was Cambrian and Ediacaran new fossils, exceptionally preserved fossils. Number one, and in second place by one vote less was Ordovician fish in relation to the origin of vertebrates in relation to the Ordovician extinctions. That's a Paleozoic. People love their early things, so maybe we need to go deeper in time rather than just
Well,
thinking about the
we've
Mesozoic.
done a lot. We've done a lot in the Triassic, haven't
We
we?
have done a lot of Triassic.
So yeah, it's right that we address the balance by going
Yeah.
a bit deeper in time, I think.
Okay. So this week I'm going to take the lead on this topic, and I'm going to cheat slightly because I'm going to include more than one paper. But that's because they're natural bedfellows. So we're going to go back to the Ediacaran and the Cambrian, or Ediacaran, Ediacaran.
I prefer Ediacaran myself, but
Let's go with Ediacaran then. We're going to go back deep time to the origin of animals because that is a question that attracts a lot of interest, quite naturally, because it's a major evolutionary event. But what makes it really cool is that it just happened fairly suddenly, right? The Cambrian explosion is a term that people use a lot. So I'm telling you this, but you know this. For a long period of time, life was pretty simple for like billions of years, and then suddenly, about 500 million years ago, animals and loads of things turned up on the scene all at once in a massive, very rapid evolutionary diversification at the very beginning of the Cambrian. It's what defines the era of life, of animal life, the Phanerozoic, right? So you're well familiar with that, and well, I was going to say you might teach that, but you don't do any teaching, do you?
oh, that's outrageous. I do. I had to
Oh, you
teach
do teaching at Birmingham,
a
don't you?
whole
Yes.
lecture. I had to teach a whole lecture for one of the museum's MSc courses. And do you know, Rob, I've got some
Yes.
marking to do. It's terrible. I don't know how I'm going to cope. Oh,
One lecture. Well, I hope you ever
my,
lie down after that, that exertion.
it was
Two.
two. It was
Two.
it was a full hour and a half of content, two 45-minute lectures, and a practical.
And a
I will
practical.
have you
Okay,
know,
I'm sorry. I'm
it's
sorry. Uh, but
outrageous.
but you don't teach the Cambrian explosions, I'm guessing. I teach the Cambrian
No,
explosion quite a lot. So, but also I go into conferences and hang out with these Cambrian and Ediacaran types quite a lot. Did we?
and I know, Rob, that in
Yeah.
honor of that, you are wearing your Anomalocaris
I've got
t-shirt.
my yeah. Here he is.
Very
There,
nice.
there he is. I
I
have my
do my
Nomlocaris
best.
t-shirt on from given to me by my sister. Okay, so that's the context, right? So it's a really interesting evolutionary event. Like, it raises so many important questions. Like, why did it happen then, after a long period of nothing happening? And also, if all these animal groups diversified very suddenly, why? What? How? How and why did new diverse body groups arise? Question. You've
But
got a question.
I've got a question. I've got my hand up for the benefit of the listeners. So we had a discussion about this on one of our episodes. I forget which one, and we were talking. I was asking about Ediacaran fossils. We were talking about Ediacaran fossils because you were saying that they were now thought to be animals, or at least some of them were thought to be animals. And I was saying, well, how do I mean? I guess we'll probably get into this, will we? But is it so? Do we now think that the Cambrian explosion is like the diversification? It's not the evolution of animals; it's the diversification of animals. Is that right?
Yes. So we need to get to that point, right? So all this things happening at the Cambrian, but actually, as you quite rightly pointed out, just before the Cambrian in the Ediacaran, we see some tantalizing impressions of stuff happening, right? There's always been a bit of a mystery. It's always been a challenge, right? To do paleontology at that kind of time, you need to do all sorts of really weird stuff. You can't just get out a bone, put a brush on it, and put it in a phylogeny. You need to think carefully about what it is. Sorry, was I just dismissing what you and I do? You looked a bit aggrieved at the idea that that's all paleontology. It's just put a bone, put a phylogeny, done, end of.
Well, we have many bones in one specimen. You just have smudges on rocks.
How dare you? Sometimes they have bones. Sometimes they don't. Sometimes it's even asking whether it is or isn't the skeleton is a problem, as was last week's episode. Anyway, so in the Ediacaran, we've got this really weird stuff. Now, what makes it so we've got all these interesting things in Canada, in Russia, in Namibia. We've got these deposits from around the world. Australia, I'm forgetting Australia was a really important one. We've got these really interesting deposits where we've got these tantalizing impressions of stuff that's very difficult to look at. Not look at. That's my eyes. What is it? No, these little fossils, they look a bit like seaweed, they're algae. Some of them have got a sort of a front and a back, but they don't have a head. They don't look like they have any eyes. They don't have mouths. So what are these sort of fairly complicated, sort of but simple, flat-looking things doing? Are they animals? Are they algae? Are they lichen? Most people now agree that they're some sort of proto-animals that fit early in the tree. So a handful of them might be proper bona fide animals, but those aren't really properly agreed. But the point is they're tantalizing, unusual weirdos that turn up. And in the Cambrian, we get proper bona fide animals. We get trilobites, we get brachiopods, we get worms, arthropods, vertebrates. That's when you see things that you can go, aha, I know what that is, and also some stuff that's weird. So there's this big difference between the Ediacaran and the Cambrian. Now, that's the context. But what makes that extra more challenging is that you've got to really get in your head around the fossil preservation. The way that the fossils are preserved in the Ediacaran is completely different to the way the fossils are preserved in the Cambrian. I'm oversimplifying here. So that makes it really hard. So even when you're looking at the fossil, you've got to think about how is it preserved, what is it, and what does it represent? In the Ediacaran, we've got these sort of flat mats where things have been preserved in this sort of volcanic sea floor. So it's really amazing. But you've got mostly all you're dealing with is this sort of impression of what the external surface of this thing might have looked like.
So is it there? When you say volcanic sea floor, they're not preserved in lava, presumably. They're preserved in ash deposits.
Yes.
It's like Pompeii, but
yes.
600 million years ago.
It's not quite the fact that it's like a volcanic. It's more like ash might have. Well, I shouldn't have said the volcano thing. It's a fine layer of things, preserving it in life and encasing it with a sort of microbial mat. It's a very flat, simple seafloor system. Yes, let's avoid the use of the word volcanic because it makes it more complicated. It makes it sound catastrophic. It's actually, as you alluded to in the previous episode, it's quite a nice, peaceful, flat place with not a lot happening. There's not a lot of predation, just quiet, bubbling around, doing your.
So, and probably attached to the seafloor, not a lot moving, right? Yeah.
Not a lot moving, hardly anybody moving at all. Occasionally a Dickinsonia might be just flopping about, and one of them, Kimberella, might have been moving or scraping, but most of the time they just sat there doing nothing. Whereas the Cambrian is a full-on throttle, violent ecosystem.
But so how are they feeding, do we think? Are they chemosynthesizing in some way?
A lot of osmotrophy is often.
What's osmotrophy?
So this is you're just using your I can't come up with the definition off the top of my head. So you're just being really flat. They've got an exceedingly high surface area to volume ratios. So what they're doing is maximizing their ability to absorb stuff from the surface on which they live potentially. Although even that is, quite a long, complicated discussion of what these things are doing. Moving into the Cambrian, we've got now these big animals, but it's very different because then we've got a very different kind of preservation. That whole window of Ediacaran style preservation is gone, and now we've got a whole new window of type of preservation, which is that we've got these nice soft tissue preserved things that are preserved. They're buried very rapidly in fine silt, lots and lots of layers, deep sea burial, and it's all nice carbonaceous films with things laid on top. So the preservation between the Ediacaran and the Cambrian is exceedingly different. So you've therefore got to try and detach what is the evolution, how are the things that you, when you're looking at the changes from one to the other, are you looking at evolutionary changes, or are you just simply looking at preservational changes? And that's why it gets a bit of a complicated problem, which is the context for the paper we're going to mention. It's the question at the front row. Maidment, what's your question?
It's a bias in the fossil record. Is it a bias in the fossil record? It's a question.
Yeah.
So my question then is what causes this change in preservation? Is it, you mentioned algal mats. So the Ediacaran fauna are they being preserved because they've got like a layer of algae that is forming over the top of them, and that doesn't then form when animals have evolved and diversified, because they're then eating that algae, so the algae is not being able to form big long, because algal mats are a thing. I mean, like things like stromatolites, we see them, which are algal, they're structures made up of algae and sediment, aren't they, like interlayered, and we see them a lot in the Precambrian. As soon as animals start to evolve, you know, those they're getting grazed, so you don't see them. Is that
sure?
Is that the difference?
That might be the case. That's possible. I mean, we're getting a bit outside my area of expertise now, because we're now talking about geology, so I have to be a bit careful here. But the microbial mats are really important in this. So the microbial mats is what these Ediacaran animals were living on, whereas in the Cambrian, we've got a sort of normal-looking ecosystem. But one of the big changes that goes between the two of them is burrowing. So in Ediacaran, we've got this really flat ecosystem, but as soon as worms and burrowing things start turning up, then everything gets churned up. So it's possible that it's the animals themselves coming along that has changed that whole preservational potential, because rather than having a nice flat microbial mat, instead you've got worms that are making a right mess of everything, and therefore the way that things get preserved has changed. Or it might reflect some other sort of abiotic geological thing. But yes, the animals themselves are changing the ecosystem in which they live.
Cool. Thanks. Cool. Yeah.
That's what Jane is busy working on with Russell. So right now in Manchester, we had Jane on last week. She's thinking about those sorts of things. Okay, so cool. That's the context. We haven't actually mentioned the paper. Good Lord, that's a long intro. But I guess it's important because we need to understand.
That's the end of the podcast. Bye, everyone.
Bye, everyone. That's the Ediacaran of the Cambrian. So let's go back to trying to find my actual notes now. Bear with me. Okay, so the paper is The Terminal Ediacaran Tongshan Lagerstadt from South China by Jingbo Hu, published in November 2025 in Nature Communications. So Jingbo Hu and a load of colleagues from Nanjing in China, one from MIT, described this new discovery in China near Wuhan of coronavirus fame. Poor Wuhan. It's a big city with a lot going on for it. And it's just now, there's a lot happening in Wuhan that isn't coronaviruses, including this new fossil deposit. Right. So they describe a fossil deposit newly discovered, which they've been busy excavating over a large number of field seasons. And it sits firmly in the Ediacaran. So this is they've used some isotope dating and ash beds that lie above it to firmly put it in quite high up in the Ediacaran. So it's quite close to almost being in the Cambrian, but it is within the Ediacaran. It overlaps with some of the things that happen in Namibia, but it's after the famous ones from Russia and Canada, and also the ones in Leicestershire as well. So what's interesting about this place is, so the Ediacaran, we've got these fossil deposits. As I say, they turn up all over the world at various different times, and they often have sort of similar looking things in it. But what's cool about this one is that the preservation at this thing is different. They have Cambrian style preservation with carbonaceous films and nice bits and pieces with cellular preservation detail, but they have it in the Ediacaran. So that means you get to look through that window. So we talked about that it's very difficult to have this idea of knowing what is the difference between your evolutionary pattern and your sort of taphonomic geological pattern. This is an opportunity to look through it, which is why I was quite excited when I saw this, because that sort of allows you to disrupt that binary over what you're looking at. So what do they find? Well, they start off describing lots of algae, which again is something you might expect to see in Ediacaran or even earlier, right? Algae have been going on for some time.
Well, when you say some time, I mean, we're talking sort of 3 billion years, right? Give or take, plus or minus.
Give or take. So we've got some algae. They've been going on for a long time. But that
I mean, some of these things in the pictures, I would be hard pushed to know that was an algae, an alga.
Yes.
In J is just, it's just some swirls on a rock.
Yeah. swells in a rock. Some of them are branching and look a bit like a seaweed, so I'll accept that Tanagi. But yes,
Yeah.
I'm not. That's why I'm not going to.
Some of them aren't, though, are they?
No, no. That's what we're going to move on to some of these other ones now that are putative metazoans. A metazoan is the formal technical name for animals. So we got a thing that looks like like a cone, which they don't really talk about. They just call it proniconites and move on and leave it alone. They've also got this weird thing called Andromeda, which is an Ediacaran thing, which is it looks like an alien basically. It's an 8-armed swirled thing, which has been known from the Ediacaran type preservation, but here it is in a very different kind of preservation. No one knows.
It's rather lovely, I think.
It is lovely, yeah.
It looks like a galaxy, which is presumably why it's called Andromeda. It does.
Yes, and it always has 8 arms. This thing.
Are they? Do we know much about it? Are they separated arms? Is it like a mattress with 8 kind of rods, or are they separated? Do the arms do you sometimes find the arms?
Yeah, it's like they sort of like they have a thing in the middle and it sort of swells out.
Yeah.
So it's a weird thing that no one knows what it is. But then we move on to so I'm going to move on from that because there's not a lot to say about Eodromeda other than it looks nice. But then we move on to the things that are much more relevant to the origin of animals.
Oh, relevant. Sorry.
You think they're iffy? I want to go on to the fronds. So what they do find is some Burgess Shale type preservation of typical Ediacaran fronds. So when I say a frond, what the hell am I talking about? Well, Charnia is one of these most famous Ediacaran fossils. It has like a disc, a holdfast, and it sort of attaches to the surface. And then above it, it has sort of like a leaf coming out of it. But it's not a plant. It's some sort of animal-y type thing. But lots of animals do that sort of thing like today. They will have a little holdfast attached to the rock and have a sort of, I don't know, body sticking up out of it. Tunicates might do something a bit like that. There are other Nidarians that do that. So it's not weird to have an animal that looks like that necessarily.
But Charnia is cool, right? Because it's, I mean, for many reasons, but also it was, as you mentioned Leicestershire earlier, it's from, known from Charnwood Forest, right?
Yes. So
which is in Leicestershire.
Yeah, so this was when, before, a lot in the 50s, it was thought that no animals existed in the Ediacaran until some schoolboy told his teacher, geology teacher, I forgot his name. Charnia was named after him, I think. I can't remember. Anyway, he found it and he got his teacher to look at it and went, oh, actually, in the end, they found it in Bradgate Park, which is a lovely park in Leicester. I've been there. You spent time in there when I was living in Leicester. But then actually I found out through Emily Mitchell that it wasn't a boy that first discovered it. It was a girl, and no one listened to her when she first discovered it. This is an excellent story of historic erasure. And again, I don't know. Okay, in my defense, I was about to say I don't know the name of the girl, but I also don't know the name of the boy. So
it's fine. But
that's okay.
Today, the Charnwood. I mean, I went on a field trip to Charnwood Forest and measure metamorphic rocks when I was an undergrad and got to see some. But there's a Charnwood Forest geopark now, isn't there?
Yeah, there is.
I have a feeling though that there's not. You can't really see too many. They try and keep people away from the fossils, don't they?
Yeah. The people try. Yeah, it's a bit of a difficult. It's a really interesting challenge because you want people to understand about the geological heritage of why the area is special. But the more you publicize it, the more people want to go and look at it. And then people try and take the fossils off and damage it. But you can't. Ediacaran fossils like this, you can't hack them off very easily and take them back to the lab. What you have to do is
they're impressions, aren't they? They're chunks of rock.
So you end up destroying it. Even if you were successful, it's gone and lost anyway. But if you end up just destroying it for everyone forever. So it's a bit tricky to balance that. How do you do that properly? Jack Matthews has been doing some good work on that with the geological heritage there. Anyway, back to the fossils here. We've got some lovely fronds, but now they're not preserved as these weird microbial matte impressions. They're now preserved as carbonaceous films in a Cambrian style. So we've got these typically Ediacaran organisms in a Cambrian style. Lovely. Now, what does that offer us? Well, it's a bit tricky, really, because there's not a lot. They look really nice, though. So it does help. Well, I guess one of the important things this does tell us is that you can get that preservational window in the Ediacaran. We've got these lovely looking, we've got a completely new way of looking at these frond type weirdos. But the other thing that I thought was quite exciting about this was that this helps answer some questions about what isn't preserved there. If this taphonomic window, if this preservational window now stretches back further in time and you don't find arthropods and trilobites and brachiopods and mollusks, then it no longer becomes a question about whether or not we just don't have them. It's more of we can now make it a hard absence, if you know what I mean. This now is it helps strengthen the idea that there's an evolutionary step change happening at that time, and we can constrain when and how that's happening.
So these fronds, I know this is not also the point of this paper, but there's so the Ediacarans, back when I was a student, the Ediacarans were thought to be probably single cells, which I think we know they're not now, massive single cells, and also a completely extinct clade type of life, right? And then that has kind of changed, hasn't it? So that there's now the idea that at least some Ediacarans are probably related to life that we know. But there's this group that are called the rangeomorphs, is that right? That are still, and these are the fronds that are still considered to have been almost like some sort of weird alien type of life that was wiped out at the end of the Precambrian and has no representatives, has no descendants, right, on Earth today, or even in the Cambrian didn't. Is that still the case?
Yeah, well, that was the case, and that is the case, but it's difficult because to get put these things in some sort of phylogenetic study of trying to understanding where they fit in an evolutionary tree is really hard.
So that was my question then. With this sort of preservation, was there anything that came out of this, with this style of preservation that allows you to solve some of those problems?
Well, actually, Francis Dunn at Oxford has probably made the biggest leap forward with this. I mean, lots of people are working on this problem in lots of different places, but her paper was really strong, where she did this really in-depth study looking at how this thing grows, placing it in a phylogeny, and doing loads of different ways of looking at the evolutionary trees. And she firmly placed Charnia, which is, represents these frondy rangeomorphy things, as an early animal. So it's more animally than a sponge. It's less animally than a jellyfish.
Fish.
In her solutions. So that's firmly, that as far as I'm aware is pretty much the status quo. This new stuff could help with that, but one of the other things that makes it possible is that if you get these sorts of carbonaceous films, you might be able to do some chemistry on it as well in a way that you weren't able to do with the ones before. Now that has happened to Dickinsonia. So people were able to get a tiny little fleck of carbon from a Dickinsonia in Russia and did some gas chromatography on it and found that it had some sort of animally kind of properties to it. So that might be possible here. But also that was a bit disappointing because then it turned out that algae could potentially have that particular kind of fat or whatever it was. I can't remember what it was in it. So there's a possibility there. But I think that gets me most excited is that means that window exists. So you can potentially start asking about what isn't there and is there. So as these authors say, the Tongshan Lagerstätte still indicates that exploring for more Burgess Shale type Lagerstätten in the Neoproterozoic is now a practical and promising task. So they're saying that this opens up the promise of that you could find more of them. There's also, I should point out, the possibility that some of these weirdos from the Ediacaran were actually clinging on a little bit into the Cambrian. Some people have looked at these things in the Cambrian and gone, that looks like a frond. But that's the other way of looking at it, right? Rather than the ones that were the window going a bit earlier, sometimes some of these things were surviving. So that was the current state of affairs. That was what I wanted to tell you about it. Did you have any thoughts or questions or worries?
Think I've brought them all up.
Yeah. So that's the take-home message for that one, I guess, is that you might be able to get a sneak at what goes on before the massive explosive diversification of animals. And actually, the fact that animals turn up then might be a real thing, not an artifact, because we're beginning to break down that barrier between the Ediacaran and the Cambrian of being really different in terms of their preservation.
Well, so what this paper basically demonstrates is that these animals with hard parts and the ancestors of today's major clades are not present in the Ediacaran. Even when the preservation, even accounting for preservation, they're still not present.
Yeah, absolutely.
So the Cambrian explosion still stands.
Yes. It is tricky though, because how many fossils do you have to find to say that you haven't found one? How can you ever properly identify there being an absence? That's hard. As you just evidenced last week with your ceratopsians in Europe, right? Maybe you look at it, you could jig things around and it might change things.
Yeah.
So that's difficult. But this one is giving us a sort of, not just accounting, but like a stepwise change in our understanding of this. I think it's cool. So I had a second paper that I want to tell you about that goes hand in hand with this one.
The fossils in this one are more convincing.
I'm breaking the format by immediately doing two papers rather than one.
So greedy.
The second paper was by Han Zheng and Qi Liu, both builders having an equal component of this, also from Nanjing in China, with a much bigger list of authors this time. And this was published in Nature, not the other one. This was published in January of this year. So this is a Cambrian soft-bodied biota after the first Phanerozoic mass extinction.
Oh,
So
so this is the other end of this is the other side of it.
yeah, the other side.
This
So
is,
we're just
see.
gonna now
Okay.
we're gonna flip across from one to the other. We now got very typical Burgess Shale type preservation. So that's this window where we get these soft-bodied Cambrian things, where we get these nice carbonaceous stones, maybe with some minerals on top of them. So they basically discovered a new Burgess Shale. That's quite reductive to say that. So in the Cambrian, we've got all these different sites from around the world. The famous ones are Chengjiang in China, Burgess Shale in Canada. These offer these tantalizing insights into the Cambrian because they have not just the skeletons, but the soft tissues as well, preserved, captured in this rapid burial and fine preservation with all these nice silts. And these guys have basically just got, hey, we've got another one. They've placed it in between, chronologically, the Burgess Shale and the Chengjiang. So that's kind of fun.
Chengjiang's older, right? So that
Yes.
it's halfway, but and the Burgess Shale's younger. Okay.
Yes, that's right. Yeah. So they present some pretty pictures, and as you point out, the pictures are a lot prettier than the Ediacaran one. But that's not to be. I'm not. That's because that's what I would expect, right? The Cambrian things would be there. So they find in this loads and loads of pretty pictures. I mean, pretty pictures isn't the way to do it because it's an audio format, but
I mean, there's some amazing fossils here. I mean, you can see there's things that look like, proper. I mean, I'm gonna describe them as insects. Clearly, they're not, but arthropods. I don't know, but they've got legs. I mean, you can see the legs. You can see filaments. You can see there's a thing that looks like an ostracod with like shell and a head coming out and little arms. It's amazing. It's a thing that looks like a prawn. I mean, then it's incredible preservation.
Absolutely. So they've got a wide range of arthropods. None of them are insects. Oh, but they're all early arthropody type things. We've got worms with legs. We've got sponges, which is important. Well, we're going to come back to that later.
Wait, worms with legs?
Yeah.
Does that not make them not worms? That's not the definition of a worm?
I actually had a long chat with Harry, PhD student at Manchester, about what is and isn't a worm. We were trying to come up with a definition about what is and isn't a worm. It gets a bit difficult. But lobopods are walking worms with legs, yes. And they are a big Cambrian thing. That's where arthropods came from, worms with legs.
No.
Well, depending on how you look at it. I don't want to... That's a bit reductive again. Anyway, they've got all these lovely things. They've got ctenophores. They've got worms.
What's a ctenophore?
Oh, like a little jelly, but not a jellyfish. It's like a comb jelly, like a little sea gooseberry. Not a sea gooseberry, is it? Sea gooseberry?
Not a clue.
You
I'll
have to go
Google
rock
it
pooling
later
with me. Let's go rock pooling, and we'll go and find some phyla.
That'd
I like
be
doing
lovely.
that with the students. Going, how many phyla can you find in this pool? Anyway, they've got sponges.
That's cool.
They got Vetulicolians, all these really interesting Cambrian weirdos, and some of the poster boys and girls of the Cambrian explosion, which is these arthropods, the ones you pointed out, with nice legs, internal anatomy, lovely. Now, there's not much to say about that because really, they've got pages and pages of these pictures, and even more in the supplementary information. Absolutely loads of new taxa, and this is kind of weird because how can you with one paper say, "Oh, we found a Burgess Shale"? I guess this is actually what should be a whole series of papers that will go on for years of them going through each of these taxa, describing their anatomy and evolutionary implications.
Wow, they've got some nice trilobites.
Yeah, well, the trilobites is what they've used to date it as a particular time. Lots of sponges, lots of worms with legs, and predators. They've also got predators here. That's the other thing that's important. Because that's another thing that's a big change between the Ediacaran and the Cambrian, is that once two big ecological changes happening are burrowing, which we talked about before, in the sense that can make a big difference, and also predation. That's going to completely give ecosystems a kick up the arse and completely change the way that animals lived, moved, survived, all of that sort of stuff. So they did some big analyses on this. That's the weird thing about this. Yeah, so I was thinking about that chain of thought. What really this is like you've got now a career set for life. You're going to describe each of these taxa. You're going to do an evolutionary study on each one of them. You're going to think about the preservation of each one of them. That could keep you going for a whole career. But of course, you want your big splash Nature paper at the beginning. Why wouldn't you?
Absolutely.
And they compare the ecosystem of this new place, Huayuan, in near Wuhan, with all the other Burgess Shale ones, including Qingling, which was Quejiang, Chengjiang, Burgess Shale, the Wheeler Formation, Margin Formation, a lot of them in Canada and China. And they find that this new one sits firmly in between as a sort of transition between the Burgess Shale, which is this famous Canadian one, and the Chengjiang, which is this early Chinese one from Yunnan province. So that's pretty cool.
It sits firmly between in terms of what? In terms of the taxa
Well, the
that they
composition,
have?
yeah, the composition of the species involved and the sorts of format of it. Now, the slight difficulty here is that when you're looking at an ecosystem like this, they mentioned that, well, we haven't got a lot of burrowers in this one. We've got more things that sit on the surface, and we've got some predators. And like the Burgess Shale has a lot of arthropods in it, and the Chengjiang has a lot of worms in it. So, you might try and do some of these plots about ecology and say there's been a change in the ecosystem, but actually, really, you've got to be tricky here, because you might be just picking up a taphonomic signal, right?
Well, what about deposition environment? Could
Exactly.
it be that, one of them is deeper water than the other, for example? Because
Yes,
of course,
absolutely.
it's marine, but could one be like, a sort of like shallow marine and shelf environment, and one a more deep marine environment?
You do have to be very careful about this. Also, because there's transport has been involved in a lot of these, right? So some of them are like a burial of a massive sea cliff. They've been rapidly buried. Maybe they were sitting up high on the sea shelf in a nice, reef-like environment, having a whale of a time, and then suddenly the whole thing collapsed and they got buried like, a kilometer, 3 kilometers away from where they were living. And there might have been all sorts of tumbling and sorting, and and that might be happening in Canada, but maybe that wasn't happening in Australia. Maybe that wasn't happening in China. That's just talking about the particular geological conditions of where you happen to live at that time, which might not reflect anything to do with your ecology and how you lived and survived when you were an organism. So
well, this is actually, but this isn't just a problem for Burgess Shale type.
No.
Any marine community, I guess, or marine group of fossils, you have that consideration, don't you? So when people are doing ichnofacies, this is where they use the type of burrows to think about what the environment was like. You have to consider that there as well. Like, were these animals living here, or were they burrowing there because they were just been suddenly brought down here in a massive submarine landslide or something?
Yeah. So that is tricky. And for the latter half of the paper, the Han Zheng and Qi Liu spent a long time talking about the paleoecology and telling a narrative of weaving together that this was happening and then this was happening. But I think you've got to be very careful about that. But hey, why not? They've got a lovely new deposit. Why not do some arm waving? Go for it.
Absolutely.
You spent ages getting this. Now, I did notice that they're both in Nanjing. I wonder if they sit in the two papers we just discovered. Do they hang out in the same tea room? Were they talking to each other about what they were doing? I don't know.
I hope so.
Do you talk to your colleagues at the Natural History Museum?
Well, not if I can avoid it. Good God.
There's not some like horrible drama going on with one dinosaur team and the other dinosaur team.
Well, there's only two of us, so
yeah, we can't fall out.
Not really, no.
Okay, well, they're in different labs in Nanjing, so I don't know about that. They've both got Ediacaran, Cambrian, Burgess Shale type things going on, so one hopes they've got. The other thing is that they've got a whole career set up here. This second paper, but that's going to take years and years of work to work through all these things. So I look forward to hearing more about that.
Well, the Burgess Shale, people still work on Burgess Shale fossils now, right? And that was found in like when?
Yes.
18 something.
Yes, I went to the Burgess Shale for its 100th centenary celebration. They had a conference at the Burgess, well, not on top of it, it's up in the mountains, but that forgotten the name of the town in Canada near it is. It had a big fire not long ago.
Oh yeah.
It's a beautiful place. It was a really good conference. I very much enjoyed that, and got to go and stand on top of the Burgess Shale with Derek Briggs. So that was fun.
It's very exciting.
There's 100 years of studies, and it's still going strong, right? So there's still new discoveries being made there, but also because people are finding sites nearby that are subtly different. The Burgess Shale is one of them, but then there's like a few valleys along. There's I've forgotten the name of it, but another one that Burgess Shale but different. And the same might be happening in China sometime soon. I guess China is seeing a lot of this because, well, China's becoming a paleo powerhouse. It's a big, diverse country, but also they're actually got so many scientists actually out there busy investigating it, and they're properly rewarding their scientists to go and do these sorts of things. So there's been a big change in the way that science in China is being done in the last, 40, 30 years. Do you ever do any work in China?
I do, yeah. I mean, I worked a bit in China quite early in my career during my PhD. My first data collection trip was to China. Went for a month to Beijing and then out to Sichuan province and Chongqing. It's pretty tough, back in 2004. No Google Translate, just traveling, no internet. I didn't even have a laptop. So I was traveling with a Lonely Planet guide. That was it. Well, and Richard, me and Richard Butler went together.
Richard Butler was your Lonely Planet guide.
Except I tell this story quite a lot, actually. He'll be really pleased to hear me tell it again. Like before we were going to go, we thought, because back in the day there wasn't Google Translate. He said, there's a language course in Chinese. It's a free intensive 3-week intensive course in Chinese that we can do before we go. And I was like, well, that's a really good idea. We should definitely do that. Just to that will really help. So I signed up, and then the day before the course started, I said, are we going to this course this evening then? And he goes, oh, I didn't sign up. So I did a 3-week intensive course in Chinese at Cambridge, also, where everybody else in the entire course spoke already like 6 languages fluently, including like Korean and Japanese. And I like clung on by my fingertips for 3 weeks. Actually, I didn't even go to the last 2 because I just can't do it. I just can't do it.
So how many a day?
Hard.
So it was intensive. How many hours a day were you doing that?
I think it was 2 hours in the evening every evening for 3 weeks.
So this is an interesting coincidence there, because when I was an undergrad, I also signed up for a Chinese course because I was interested in China and whatnot, and it was like 1 hour a week. And I had the same experience of you, right? I went along with a mathematician friend of mine, but he and I gave up really early because about 3 weeks in, the teacher took away our pinyin and said, "Now you're doing the Chinese characters now." And everyone else in the class already spoke Japanese or Korean or some other language, so they were like, "Oh, let's go!" And I'm like, "Oh, good lord." So I don't feel good about that. I'm gonna stick in my French and Spanish lane, and
yeah, quite.
Chinese I found too hard. I couldn't do it.
It is challenging, but these days it's so much easier to travel everywhere, isn't it? Because you get.
Yeah.
I tell my students they don't know they're born. Honestly,
at the risk of being an old fart now, I have to tell the kids that if you wanted to go, if you were traveling somewhere and you had to have the maps, timetables, and books physically with you, otherwise you wouldn't get that information. And that makes me sound it's like a nearly dead person. Can you imagine a time where you wouldn't just look it up on your phone and immediately know where to go?
Exactly. Anyway, yes.
Anyway, this is now turning into old farts reminisce. The Chinese paleo powerhouse. Look forward to more of that.
Yeah, absolutely.
Take home messages from these is get digging, right? Because there's new stuff to be found out there. But this is serious. Each one of these 2 papers has been lots and lots of field studies and exploration of what's going on. I didn't actually explain where in China the Huyan was. It looks like it's in between Chongqing and Wuhan again. So it's in the center of China.
And I think the thing with China is that, you said it's a massive country. There's loads of paleontologists working there. They're really well funded. But also, I mean, it is undersampled compared to, say, North America, Canada, Europe. Yeah. So although, paleontology has been big there for, well, I mean, since the 1930s and really got a big renaissance, I think, in about the 1990s, maybe the late 80s, early 90s, it's still, because it's so huge, it is still undersampled. There are still amazing things to find.
One imagines there might be some sort of tail off. I don't know, but China is massive. I mean, you're not going to find a new Burgess Shale deposit in the United Kingdom, I don't think, because pretty much every corner of the island has been picked over. Although that Lucy Muir and Boating, they go poking around in Wales and they occasionally find some new soft tissue preserved things. So there are new discoveries being made, but the biggest gains are to be made in places that haven't been properly explored.
Definitely.
Okay, so have we got time for a final paper?
Go on, hit me with these sponges.
You could sound a bit more enthusiastic.
So sponges, just to be clear.
Yeah.
How do we know that sponges are animals?
They have tissues.
Not plants.
They have tissues. So, I didn't actually answer the question about what's the official definition of an animal. So I guess I need to talk about that. Well, it's a bit tricky. There are a number of different definitions, but you've got to move around at some point in your life cycle. So actively locomoting is one of it. Now, lots of animals don't do that, but they do it when they're a juvenile. But that's also related to having tissue. Do you have some sort of extracellular matrix? Can you move your cells around your body? Well, no, that's not true, because being able to move your cells, plants don't do that, fungi don't do that, right? You have a cell, the cell stays where it is, it stays where it was. Whereas if you're an animal, you can move about and have a bit more locomotion related to that. Yeah.
But how is something spawning into the sea?
Yeah.
And those larvae things drifting on currents and then settling different from pollen in the air?
Cilia. They use cilia to move themselves along.
Oh, do they? Okay, all right, fair enough.
Yeah. I may be wrong about this. I look forward to some fossil listeners telling me what the proper definition of animals is. But having cilia and moving about and having your cells and an extracellular matrix is an important part of that.
I don't understand what an extracellular matrix is.
Oh, so no, I can't explain that off the top of my head. I'm gonna have to get stuck. Now, I often hide behind the fact that you're the geologist and I'm the biologist, and therefore I can't answer that. That's a geology question. But even just supposed to be a direct biology question, I'm now chickening out.
I mean, it's a broad discipline, Rob. You can't know everything.
So having a whole lot of proteins that are outside of your cells. So if you think about a plant, right, you've got all these cells with walls and it's all like a big block. But animals have all this stuff going on outside their cells. Okay. So there's all this matrix of things moving about. So that allows things to move in and around and about it. So the final paper is Independent Origins of Spicules Reconcile Paleontological and Molecular Evidence of Sponge Evolutionary History, published in Science Advances in January 2026 by Maria Eleanor Rossi and a whole bunch of people from Bristol and others, including Joe Keating, former collaborator of mine, and my former PhD supervisor, Phil Donahue. So these guys love their fossils, and Davide Pisani he loves his phylogenomics. So I'm going to have a very quick version of this because this is just to tell the story. They did a big phylogeny of sponges. So sponges, we talked about before, they are animals, but they don't really, they make it a real challenge to define what an animal is or isn't because they don't have a brain, they don't have a head, they barely have any kind of organization of tissues. They're just like a filter feeding blob that sits on the surface and uses little cilia to eat out all the chunks that are inside the sea. So presumably you're familiar with sponges.
Well, I'm familiar with, that you wash with them. Also, though, also there are there are sponges that do have a skeleton, aren't there?
Yes.
There are glass sponges and sponges that we find sponge spicules in the fossil record as well, don't we?
And that's exactly what this is about, right? So sponges are one of the earliest branching animals. I'm not going to actually this at a risk of going into a massive debate here. There's been an enormous blood-spilled academic debate about whether sponges or ctenophores, the sea gooseberries, the comb jellies that I just mentioned earlier, are the sister group to the rest of animals. And the amount of phylogenomic arguing about that is crazy. Also, because the evolutionary implications are massive. But we're not going to go into that today. That's a whole story for another time. Really interesting story. If you love your phylogenies, which we both do. So anyway, they did a big phylogeny using the genetics of sponges. So they sampled a whole load of sponges that are alive today, compiled loads and loads of genetic information, did a big phylogenomic study of it, included some fossils to calibrate it and think about when this was going on. And they were trying to figure out what the earliest sponge would have looked like. Would it or not have had these spicules? Would it have had a skeleton? Because as you're pointing out, sponges are these, well, we think about these like sacs sitting on the bottom, but a lot of them have either silica as a little protection, spiky bits on the outside of it, or calcareous, spongy, spiky bits. And actually, because we don't really probably know the earliest condition, what did the first sponge look like is a really important question for this. We're thinking about the Cambrian Ediacaran. So they did this big fancy analysis and demonstrated that actually the earliest sponges didn't have a skeleton. So that was the thing that we didn't really know before, that they were actually able to demonstrate it quite firmly, that the earliest sponges were entirely squishy and didn't have spicules that would be preserved as a fossil. And that is importantly relevant for the Ediacaran Cambrian thing, because a lot of these studies looking at genetics and DNA imply that there's an enormous missing record, that we should have animals occurring in the fossil record much, much, much earlier than we do now. Now, there are all sorts of really important analytical reasons why that might be that I'm not going to go into because it boils down to mass modeling and phylogenomic studies. But this is a simple ancestral state reconstruction in some ways, just demonstrates that the spicules came along later. And that actually neatly ties up the gap. That gap that we have for where sponges should turn up but don't is now quickly accounted for by saying it might just be because we don't preserve them because the spicules weren't there, which is kind of fun.
So do we see things that we think as sponges or stem sponges or early sponges in the Ediacaran? Or do we not know?
That makes it a bit this actually, I'm trying to think about how to answer that question because it gets a bit contentious. Like what is a sponge? If you don't have a spicule, how can you recognize a sponge? So the spicules are these tiny little mineralized things, but if you don't have those, a sponge is just an amorphous blob. How do you say something is or isn't a sponge? One way to do it is looking at the biomarkers, and you could say, okay, I've got some particular chemical signature that implies it's from a sponge. But that's led to a series of papers where people were arguing with each other. Some people may have been thoroughly discredited in that whole debate. I won't name any names. But anyway, Intriguing. I can't go around naming names. But there was one person that was adamant that there's absolutely no sponges, never, ever, ever, and all this evidence is wrong. And then other people came along and said, actually, you're wrong, and here's why, because you don't understand what you're talking about. But I can't name names because it gets a bit too specific. But there's drama in the world of sponges.
Sponge drama. So sponges today, I said I'm familiar with sponges because we wash with them. So those things that we wash with, they are actually sponges, right? They are.
Well, most of the time now you'd be buying a synthetic piece of plastic, but
not the synthetic ones.
I think you're telling your age there a little bit. They're called sponges because you might have actually had to buy a former spongy bit of a skeleton of an organism. Yes.
But when you go to when you go to Greece or something, you can buy So anyway, are they, because they don't have skeletons?
No.
So have they lost their skeletons, or is that just part of a thing, or what? Like what?
That would have been the spongy. I think it's spongin, maybe. I don't know.
Oh right. So that's not the whole thing.
No.
Okay.
That would have been just the sort of soft bit left behind. You wouldn't want to wash with spicules. You'd be scraping your skin out. Or maybe it's good for your exfoliation. I don't know.
But that was my question. Like the thing that we know, I mean, like for most people who don't spend a lot of time looking at sponges other than next to their bath, they don't have a skeleton. It doesn't look like they have a skeleton. But those bits it's just that we've taken off the hard bits, right?
Yeah,
it's not like that there are other groups of skeleton that have secondarily reduced their skeleton to become soft.
Well, that's exactly why this study from Bristol was necessary, because some sponges have these spiky spicules and some don't. So if you want to figure out what the earliest sponge looks like, you need a good evolutionary tree to reconstruct that, because otherwise, and that has massive implications for your understanding of the timing and origin of early animals. So that's why you need to do exactly why you need to do a nice phylogeny like this and an ancestral trait reconstruction, because it really matters for your interpretation of when animals first turned up. Was it a spicule? Was it a skeleton? Was it an animal? Was it a sponge? Was it a... Oh God, there's so many questions associated with this. And also, as talking to you about this, it implies there's an awful lot of drama and academic dispute going on around this area. Who'd have thought that the water sponge is going to be so acrimonious and
quite
dramatic?
So I had a question when I first opened this paper on the train this morning, when I was looking over it, and I was thinking it feels quite niche. I was thinking that you start off your career in paleo and you go, sponges. I am going to study sponges. But now you say it, I see why sponges have a very critical role to play in
two things to think about.
Understanding everything.
They're a major important component of ecosystems. So they're a really interesting group. But if you want to understand the origin of animals, then you need to understand sponges. And Maria Eleanor Rossi is busy doing that. But also, it's a really interesting phylogenetics question. Maybe you're not actually interested in sponges. Maybe you're just interested in how to build evolutionary trees and the data to do it, which is, something that I'm sympathetic to. And I think you are too, because you love your phylogenies. So this is, an important way to solve evolutionary problems, is build a nice evolutionary tree. So go and do it. Crack on.
Very good.
So moral of the story for all these things together, the take-home message is there are squishy things out there. You might have originally been a problem trying to figure out, do we know that they're there or not? Because the taphonomic windows, the preservation windows are changing. But we have this idea that the squishy things would have come first, and we're now beginning to get an idea that we could potentially find out if they were or weren't there before the Cambrian. And we might be able to find a squishy sponge, using this Burgess Shale type preservation in the Ediacaran. So I think there's potentially, this implies that this is about to get blown open with all sorts of new discoveries. There we go. That's Paleozoic new squishy stuff for you.
Nice.
No dinosaurs today. Although you've got the dinosaur on your necklace there, so you're bringing the dinosaurs to the table. Are you now feeling happy with this, or do you feel like I've dragged you too far down the tree of life into a murky, controversial mess?
Well, what I'm going to go away and do is Google some things. They are going to include Googling pictures of sea gooseberries.
Yeah.
Also, I want to know more about sponges. That's what I've come away with. What do sponges? Sponges. Yes. I want to know more about
sea gooseberries and ctenophores are far more interesting than sponges, I would say, because they, the question about whether or not they have an anus. It's a really interesting academic debate. There was some interesting debate about that.
Do sponges have an anus?
Sponges do not have an anus, but a comb jelly, a sea gooseberry, does have an anus.
Sometimes. When it's okay, hang on. Wait. I feel like we're getting off track here. But how do sponges excrete waste if they don't have an anus?
They're just filter. They just have the water coming and the water comes out. That's it. But that's not.
They're just like siphons.
Yeah.
But that doesn't count. That doesn't count as an anus then?
No, because they don't have a gut.
They don't have a gut. Do they have? Oh, okay.
You're looking quite panicked about sponges now. Are they challenging everything you thought about animals?
Yeah, I think yeah. So they don't have what do they have? They're just basically a bag of cells.
Yeah. Well, with a spongy silicary thing in the middle, yes. I maybe should have spent more time explaining what a sponge is or what a sponge isn't.
Well, it just sort of, strips you right back to, as you said, what is an animal? What are they doing? They're just, all they're doing is just there to reproduce, to just, but then that's all we all are here to do fundamentally, right? But
it barely counts as an animal. Sponges are not living an examined life. They are living the unexamined life. They're not spending a long time worrying about where they came from.
Must be quite peaceful.
But that's why ctenophores and the gooseberries, sea gooseberries are interesting, because
are they wondering where they came from? Maybe they are.
They don't have a brain, but they do have nerves. So the implication is, did the nervous system evolve twice? This is the big questions that we're talking about with these sorts of phylogenies.
Wait. Can sponges feel pain?
No, they don't have any nerves.
Wow. Do corals have nerves?
I don't know, actually.
What about jellyfish?
Jellyfish do have nerves, yes.
Okay, good.
Does coral have nerves? I'm going to have to Google that now. I don't know.
Okay, well, we've got,
I think, family as a jellyfish. We've gone off topic a little bit, but I guess but this is boiled down to, this is why, okay, let's now pretend that we've got a whole narrative arc that we constructed this episode around, rather than just stumbling around. At the beginning, you asked me what an animal is and what an animal is not, right? Now we can see that animals came along around 550 million years ago, but they might have not had any spicules. They wouldn't have had a skeleton. They'd have been squishy, and they would have been. All those questions related to that are really contentious and difficult, and have massive implications. And we need a good fossil record in order to get a handle on how these things are preserved. So, narrative arc completed.
Well, Rob, I think you've
with a lot of mess in the meantime, talking about intensive Chinese lessons, what an anus might be. God. Okay, thanks, Susie. See you next time.
See you next time, Rob.
The Fossil Files was recorded by Robert Sansom and Susanna Maidman.
We want to hear what you think. You can let us know your thoughts on the papers that we have discussed, and you can make suggestions for papers for us to talk about on our social media channels.
You can find our email address and links to our Instagram, Blue Sky and YouTube channels on our website, fossils.libsyn.com.